Okay, so good morning everyone. Today's lecture is going to be about tertiary protein structure prediction. So we're going to start by explaining what's the thermodynamic hypothesis. So this is a hypothesis that was formulated in the 70s, and it stipulates that the native structure of a protein corresponds to a minimum of free energy. Alright, so the idea is that the protein will adopt this structure in solution because it is a stable structure. It's and not only it is stable, but it is the most stable one. So an interesting fact is that the free energy difference that we expect between the native state that we're looking for and the ensemble of denatured conformation is about 20 to 60 kilojoule per mole. So that's 15 kilocal per mole. So that's very small. We're looking at a difference which is extremely small. So we don't exactly know how the different possible conformations of a protein populate, let's say, the landscape of free energies. But there are hypotheses about what it looks like, what this landscape should look like, okay? So hypothesis is that the structure, the native structure, should be unique. The sequence does not have any other configuration with a comparable free energy. Another hypothesis is that it should be stable. So small changes in the surrounding environment should not completely make it switch to another structure. And it should be accessible kinetically, so the folding of the change must not involve highly complex changes in the shape. Like, for example, knots. So the energy landscape can be defined as a multidimensional hypersurface, which is obtained by mapping all possible conformations of a system and their corresponding free energy levels. So if we take the example of cyclohexane, which is very small compounds with a few atoms, we can simply define a collective variable, which is the bond twisting, and this will allow us to completely describe the energy landscape of the compound. So that's what's shown here. You have the relative energy. So free energy is always relative. They don't mean anything in absolute values. They're with respect to something. So here the reference is the native structure, the minimum, which is at 0, and then you just have positive values for the other conformations, right? So what you can see is that there is a configuration corresponding to this 0 energy, which is the most stable one, which is the C configuration, and then you will have some peaks that are associated to different other conformations. So the most unstable one is the TC, and then you'll have B and TB, which are kind of intermediates, okay? So there are different bond twisting values that correspond to a given conformation. Conformation can be obtained with different bond twisting. Essentially, it's the same shape and the same energy. So here we can describe this fully. But for a system of n atoms, n being big, the number of degrees of freedom being 3n, so even bigger, generally, we do not have access to this energy landscape. So what we do is we represent only a section of the hypersurface along some reaction coordinates. So here the bond twisting is really useful, but for a big system, it may not be obvious to find a reaction coordinate, something that really describes well all the system. So there is a complexity associated to the objects we manipulate, which makes this energy landscape difficult to access and to characterize. So there are hypotheses about its shape, which is thought to be funneled and rugged. Okay, so funneled means that you're going from some conformations that are not favorable with high energy to the native structure like this, going down progressively. So there is a kind of gradient of energy which will lead you to this position, this well. And rugged, meaning that you have a lot of local minima. So this also prevents you from finding the global minimum generally, because you can be easily trapped in this local minima. So this is a view of the mind, if you wish, but there has been experimental evidence that did not make this hypothesis or this view invalid, that haven't made it invalid so far. So one may think of a protein as being an equilibrium between different conformations. Okay, so for example, this is a kinase domain, which can be in an inactive autoinhibited form, so that's from X-ray, or active phosphorylated. And between the two, you can see that the highlighted regions in orange and magenta have drastically changed their conformation. So they just open and detach from the protein, completely different shape and completely different secondary structures. So in between, you may predict some inactive intermediates by molecular dynamic simulations. We will come back to this. And there are also structures that were solved with different inhibitors, and with a structure of a mutant, right? So inactive structures with inhibitors and of a mutant. So there is an equilibrium of these conformations and a change in the environment. So for example, the binding of an inhibitor, a mutation, phosphorylation can just change the proportion of these different conformations in solution and the probabilities of existence. So the protein solution is viewed as a statistical ensemble. So here you have the basic formulation for the free energy, Gibbs free energy, which is a difference between the enthalpy of the system and the entropic contribution. So the entropic term corresponds to internal interactions, and the entropic contribution corresponds to the hydrophobic effect and also the conformational entropies, so meaning the disorder, the content of a variation in the shape. So the numbers we're dealing with here are big, right? So you have these arrows representing the extent of enthalpy we can have. So the enthalpy will be favorable. The entropic, the hydrophobic effect will be also favorable. And the conformational entropy will be unfavorable. So it's a positive contribution. We are interested in having the lowest energy as possible, meaning the more negative. So what you can see is that these arrows are big. And when you sum them up, they become something small. The output, as we said before, the delta G is very small. So this is a big problem because we're going to have some errors in the big numbers, and these errors are going to hopefully cancel out, but most probably accumulate and result in big errors on a small number, which is our target. So amino acids of a protein are joined by covalent bonding interactions. We saw this. This is the primary structure, and the 3D fold is stabilized by non-bonding interactions. So these are electrostatic interactions, hydrogen bond interactions, van der Waals, hydrophobic interactions. So when you look at the numbers corresponding to the energies of these interactions, you can see that they're small, but they are the same order of the free energy, right? 5 kilocal per mole for electrostatic interactions, when we said that the difference between denatured state and native state is about 5 to 15. So that's roughly the same order, but we have so many interactions in 3D structures. And these interactions can be considered differently if they are local, between residues that are close in the sequence, or non-local. So what's a typical modeling approach? Well, we'll choose first a representation of the object, right? So we define the resolution, the forces, maybe we use extra data. Shall we represent all the atoms? Shall we have a coarse grain representation? Then we have to choose or define a scoring function, which will allow us to estimate the probability of the models that we predict, that we sample. And we need to define a search strategy. That is, how are we going to sample the space of all possible confirmations? And usually, the final step is to refine, to filter and refine some pre-selection. So, what do we have as choices for representation? We can represent all the atoms, that's on the left, or simplified sidechains, or omitted sidechains, just the C alpha. So of course, when we go from the left to the right, we increase the speed, but we decrease the precision. So this is an all-atom model with all the atoms represented as spheres and colored according to their elements. Then we may go for a coarse grain model where we have just merged some atoms into pseudo-atoms that should keep some physicochemical properties of the initial atoms. Or we can go for something even more rough, more simplified with spherical harmonics, where we just have a shape which can be colored. So conformations can be represented by Cartesian coordinates. The constraints of the polypeptide chain must be explicitly addressed in this case, so the bond length, bond angle, torsion. We can use internal coordinates, and here we do not need to represent everything because the bond angles and length are going to be fixed. Okay, so the thing is, with this coordinate system, a small change induces a global adjustment of the downstream residues, so it may be more unstable. We may use interatomic or interresidue distances, so collective variables that reduce the number of dimensions and that we need to carefully choose. And we may use regular lattice, so something very basic. It's impossible to represent a real protein structures on a lattice like this, unless the grid spacing is very small, but these models have proven useful. So here is an example of a regular lattice. You just have, a fixed spacing of the grid, and you put the dots that correspond to residues here, and they're just colored if they are hydrophobic or polar. And so the game here is to try to bring all the H hydrophobic ones together to maximize the interactions between them. So you can have this in 2D or in 3D. So you don't have realism here. You don't represent helices, for example, but it's still useful for the folding. You can also use irregular lattice, for example. Okay, so then I go to the search strategy before the scoring function. So how are we going to sample the space of conformations? There are different case scenarios. The first one is we know a structure of a closed homologue of our query protein. So in that case, we have the assumption, we know that the structures diverge less than the sequences, right? So we're going to use comparative modeling, meaning that we take the known structure of the homologue as a template, and we're just going to thread, if you wish, or we're just gonna use the constraints in the template to predict the structure of our query protein. So for this, we need a mapping between the template sequence, the sequence of the template, and the sequence of our query protein. And we do this with a sequence alignment typically. So if we have more than 30% sequence identity, we are confident that the resulting model will be realistic. Okay, so that's just the pipeline, what I just said before. So here you can see that the choice of the template has, of course, some impact on the results. So this is typically a representation of sequence identity between the template and the query, right? And you have schematic depictions of pairwise alignments, sequence alignments. So in the first case, you have reliable hits, you can predict well. In the second case, you're in the twilight zone, around 30, and there are some residues that are identical in red and some that are not in green. You may have a few gaps here and there, so you will have to make a selection on the models you predict. There is some ambiguity that arises from from the sequence alignments. And below 10, you cannot find hits. The alignments are just too poor. So So here you again have sequence identity against structure similarity measured by the GTD-TS, which is a classical measure to compare, to assess the quality of a 3D model. So again, we can define this strip corresponding to below 30% and above 30% sequence identity. And you can see that the slope changes between these two regimes. So there is a good correlation between sequence identity and structural similarity. But below 30, you can really have drastic changes in great variability. Okay, so main message is you shouldn't go below 30. If you are below 30, it will require manual adjustment of the alignments. So there is not only sequence similarity that matters for the choice of the template, you should also consider the quality of the 3D structure and also the physiological condition, the conditions possibly in which it has been solved, the environmental conditions. Because you may have some proteins that are homologous, but they live in completely different conditions. Some of them are, I don't know, live in very high temperature. And this may really mess up your modeling. So here is an example of the impact of a gap. Okay, we have this alignment and two possibilities for it. Either we put the gap before the secondary structure, which is a strand, or at the beginning of it. So if it's before, it will just be in a loop. It doesn't have a strong impact on the structure. If it's inside, then it will just break the structure. So you may incorporate some predictions of secondary structure to improve your alignment. Okay, so then how do we build the backbone when we found a template? So there are different methods to do this. Historically, here I put some methods that date back to the 80s, which is rigid body assembly. So here you just define a conserved core, which correspond to regions that are really well aligned. And then you're gonna build up from these conserved core regions to the whole protein by adding some variable loops and lateral side chains. Segment matching, here you use positions derived from the alignments as a guide to find matching segments in a representative database of all known protein structures. Okay, so there is always this also choice that you have to make between an ab initio construction and construction that is based on what is found in databases of structures. So a very popular method is modeler, which was developed in the 90s, where you have spatial constraints that are derived from the selected templates, from the 3D structure of the templates, and thanks to the alignment, you just impose them on your query protein. They are transferred to the sequence alignments, and so the structural model that you will obtain guarantees minimal violation of the constraints, and it's a global optimization. So globally, there is minimal violations. So it's not been very much updated since the 90s, but there's a more recent paper here that I cite. So building the loops is a big problem. You may have loops that are really variable and they don't have any template, and they can be very long, and well, they are probably also very flexible. So what does it mean to put them in this particular position with respect to another one, and how do you do this? This is really still an open question. So generally what people do now is to search databases of loops and try to refine the loops with molecular mechanics, molecular dynamics. You also have to build the side chains when you have the backbone. So here, of course, it's easy if the side chains are the same between the template and the query protein, but otherwise you have to perform ab initio modeling. So you may also go look into databases to see what are the most probable rotamers, and then combine these rotamers. So here we have an optimization problem where you want to fix, given a sidechain that is fixed, then you need to find the configurations that are probable and that are also compatible with it, not to produce some steric clashes or some repulsion. So how do we validate the model? What can we expect? Well, if you're above 90%, it's as good as the X-ray structure. If you're between 50 and 90% sequence identity, the RMSD should be around 1.5 angstrom. If we're below 25, lots of errors. Sidechain stacking may be wrong, distortion, shifts in correctly aligned regions, error in regions lacking templates, errors from incorrect alignments, or just the wrong template, so not even the same fold. So the alignment matters, as I said before. Here you have an example where between the target and template there is a big gap, right? A big insertion of the template, and different method will give you different things. So typically the question is, do we join these two parts that are separated by the gap or not? And if we don't, well, if we try to do it, it may completely distort the other part, like in the A example. Okay, so that was the easy task. Now we may not know a structure from a closed homologue, but we may know a structure compatible with a sequence. So here we go from sequence to profile. So we try to generalize what happens if we don't have a high closed homologue for which we have a structure. Then we're going to look for further away homologues, remote homology, and we're going to use for these sequence profiles. Okay, so we talk here about fold recognition instead of comparative modeling. So the task is to recognize the most likely folds the query protein adopts. There is this question of fold. Okay, if the homologue is closed, you don't have the question. You know the fold. It's the fold of the closed homologues. But if you don't, then you have multiple choices between multiple folds. So you enrich the sequence description of the query protein by using probabilistic models, profile with PsyBlast, HHBlit, Jack, HHMER. Many tools allow you to do this. So this is a possibility. Another possibility, which is orthogonal to it, is to directly evaluate the compatibility between the query sequence on each of the known folds without trying to identify hits through sequence or profile comparison. So here you just thread the sequence on the structure, and you're going to evaluate the energy of the model that is given by this threading. So you can do the threading on the whole fold or over fragments that are later assembled. So here is an example: protein A threaded on template. You have some core secondary structure segments and some loops, right? And then you're going to try to you're going to try to refine the loops. And on the right you have another protein which is also threaded on the template. So A and B will be different, and the differences are mainly in the refinement of the loops. So given a protein sequence and a single template, you want the best alignment of the sequence to the template. So here is an algorithm where you represent the threading as a vector T, where each element indicates the index of the amino acid placed in the first position of each core segment. Okay, so these points that are consecutive, the tips of the arrow, even the I, J, K, L, these are core segments, right? And you're just gonna indicate the index of the amino acid at the first position of the core segment, and then all the rest will be implied. And then still you have this combinatorics problem where you want to join the different core segments together. So for this you have the lines. So a set of potential threadings can be represented by bounds on the first position of each core alignment. Okay, so I have my T, which is my set of threadings. Each threading is vector T, and it is defined by each T, I, J, K, L. So the first position of the core segment is bound by B, I, D, I, B, J, D, J, etc. Okay, so again, you're trying to assemble this. So the bounds are defined by the likeliness that you have, and also by the compatibilities that you have. So finding the optimal threading is an NP-hard problem. So how can you solve this? What type of algorithm can you use? You can use a branch and bound algorithm. So a hypothetical branch and bound search will be: each circle illustrates the space of possible threading, solid lines indicate splits made in previous steps, dashed line indicates splits made in the current step, and the numbers indicate the lower bounds for a newly created subset. So in branch and bound you have these lower bounds which allow you to stop the search at some point. And errors show the set that has been split. Okay, so this is a typical thing you can do. 2 critical issues: computing the lower bound and determining how to split. Okay, so these are orthogonal approaches. Like I said, either use sequence profile or you just don't care and you just try to find the compatibility with a template. Or you may combine the two. Profile comparison combined with threading to select a subset of potential folds and also to guide the threading, okay? Because that's also interesting to use the multiple sequence alignment to guide the threading. So structural annotations predicted from sequence, like solvent accessibility, secondary structure predictions, can be added to refine the alignment and or to help evaluate the compatibility between the fold and the sequence. So for example, iTaser is a popular method that does this, where you have structure assembly, a first structure assembly, clustering of the structures, then reassembly. So typically these methods proceed with iterative refinement procedures. So you start from the query or the target, protein with unknown structures. You search for templates by threading. 8 programs use the iTaser. Then you excite continuously align fragments from template and you build the unaligned region from scratch. You reassemble the structure. You identify lower energy models and you minimize and you assess the quality. Another very, very popular method is Rosetta. So Rosetta is a really big thing that's been built over the years. So here, the basic algorithm is you take your protein sequence P. For each window of length 3 and 9 in P, you assemble a set of structured fragments. So M is the initial structure model of P, fully extended conformation associated to a score. And you iterate until you stop, you find you fulfill the stopping criteria. You randomly select a fixed width window of amino acids from P, randomly select a fragment from the list for this window, and then you just switch M prime equal M with torsion angles in window replaced by angles from fragments, and you recompute the score. And you use a Metropolis criterion to decide if you accept or not this new conformation, this new structure. So the Metropolis criterion typically is if the energy is better, I accept. If it's not better, then something is missing here. I'm sorry about this. So then you will use a formulation with you will use a Boltzmann factor with the temperature. So basically you decide to use a parameter which is temperature. If you increase the temperature, you're more relaxed about the choosing criterion. You will accept more unfavorable structures, let's say. Okay, and then there is the hard part where when we know absolutely nothing, what are we gonna do? Then we need to do ab initio predictions. Okay, so ab initio modeling consists in searching through the conformational space of the protein with some priors about the physics, the chemistry of this object. And well, you're gonna try to just move in this space according to the scoring function that you define. Okay, so we can have a naive approach, say I'm gonna explore it uniformly, but we know it's not the case. The energy landscape is not a golf course model. So it's rugged, funneled, that's the hypothesis, and we're going to try to exploit these properties. Okay, so an example of systematic search. First, very naive, search for the optimal combination of phi and psi torsion angle values. So with the bond length and angle that remain fixed, and also the other torsion angle that remains fixed without side chains. We rapidly have a combinatorial explosion, right? So if we take a delta, the angle, an increment of 30 degrees, with n equal 2, we end up with 144 conformations. So that's a lot. N is the number of residues. With 3, 21,000, and with 5 already 430 millions. So we can't afford doing systematic search. How do we improve? Well, we can think of having a quality check on partially built structures to say, is this good or not? So like a lower bound where you just rapidly eliminate some structures. And you can think of a trade-off between the resolution of the search rate and computational time. So an alternative to systematic search is molecular dynamics. So here you are going to use an analytical expression, which is called a force field, and which represent the interatomic forces in your system. So that's represent the molecular mechanics of the system. So you need a functional to describe the interactions, and you need to choose which interactions you're going to represent. So in principle, the motions of a protein should be should be driven by the Schrödinger equations, because we are in very small scales. But to simplify, we will assume that we are in the classical mechanics, so that the motions of the proteins are just described by the Newton equation. So the sum of the force is equal to the mass by the acceleration. So this is the general form of the functional. You have some terms which correspond to the bonds, some terms to the angles, some terms to the torsion angles, and then some terms to the non-bonded interactions, which are electrostatics and van der Waals interactions. So you start from the initial model, you compute the forces, and then you move each atom according to these forces. You advance the simulation by a time step that must be very small, and you iterate on this, okay? So you may start from a completely unfolded peptide, a polypeptide, already from some initial guess. You need a numerical integration algorithm to advance at each step, and you need to have a time step that is smaller than the fastest relaxation time you have. So what typically is done is to freeze the interactions that involve the hydrogen bonds, because these have a very fast relaxation time. So you have typical time steps are of 2 femtosecond, 2 to 5, 1 to 5. Okay, so in principle, with long simulations, the entire informational space can be explored. You have ergodicity. So you may use, so you need to represent the solvent, okay? The protein is not in the void, it's in water or in the membrane, so you need to have a representation of this, either explicit with all the atoms or implicit, like a mean field. How do you set up the simulation? You choose your solvent, you froze the high frequency vibration, you need to define boundary conditions, so you can use periodic boundary conditions, you just replicate your cell at infinite, and you choose the canonical, the statistical ensemble you are working with, meaning you need to choose what are the parameters that are going to remain fixed along the simulation, knowing that you need to have the total energy that is conserved, that remain fixed all the time. So here are some snapshots of the simulation of about 5,000 nanoseconds, so 5 milliseconds. So if we look at the evolution of the field, so this is a slide which shows you it stops at 2012, but okay, it's already a long time ago, but it's just to tell you that around this time there was a kind of revolution, let's say, in the field. So it started in the maybe 2000, a bit before. We could simulate some small systems, mostly with often with implicit solvent if we wanted to access high long folding times. And so there are several groups that were involved in these things. And what you can see is that in 2012 there was a big peak with I think 12 proteins that were simulated in explicit solvent, and this was done by the David Shaw research group in New York. What they did was, first they built a machine specifically dedicated to this kind of simulations, to this kind of calculations, and so they could really fold in explicit solvent some proteins, not very big ones. But another initiative is the Folding at Home initiative, where you have adaptive sampling using the computing resources of everybody. So it's really distributed computing. You start from different points and you do small simulations, and then you try to put all these simulations in a market state model. So this allows you to compute some kinetics of the folding. Okay, so then you need to choose a scoring function. And for the scoring function, I think we will continue in the next course.